Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Max, and I'll be your host. We are delighted to have our wonderful sponsor, IDT Biologica, with us today, who'll be presenting this webinar titled Analytical Challenges in Viral Vaccine Testing. Today's speakers are Dietmar Meyer and Stefan Borutsky. Now I'll provide some housekeeping information before we start. First of all, I'd like to welcome you to our webinar platform, ON24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. During today's events, there will be plenty of time to ask questions to our speakers. You can do that via the questions widget. Just type your question in this box and click submit. We will allocate some time at the end of the session to answer questions or any thoughts that you may have. If you require any assistance, please use the help widget, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome the IDT Biologica team. Over to you.
Thank you for this introduction, Max. Dear analytical community, it is a pleasure to talk to you today about analytical challenges in viral vaccine testing and our approach to solve them by introducing innovative technologies. This is really just a small selection because there are currently a number of innovative advantages that actually require an entire webinar series. On the 1st July 1921, the IDT was founded as the Bacteriological Institute in Dessau, beginning with a fight against pulmonary tuberculosis and further animal diseases, such as anthrax, swine, and avian influenza. Today, we would call it a startup. In 1930, the name was changed to Anhaltischen Serum Institute Dessau, abbreviated as ACID, for the industrial serum production. For this purpose, a separate stud farm was founded. Directly after the World War, the production of vaccines and diagnostic material was resumed at the research institute permitted by the US military administration. In 1993, entrepreneur Helmut Glocke privatized the company as Impfstoffwerk Dessau-Turnau GmbH, abbreviated as IDT. 2016, the process development capacity were expanded by a new site in Magdeburg in Germany. Building on the long experience, we have continuously expanded our expertise in bacteriological and virological vaccines and viral vector in general. First of all, something about IDT Biologica and its vision and mission. Our vision is to be a trusted partner in shaping a healthier world. This means that we don't just see you as customer, but are passionate about your product and want to make it a success by curing and protecting against diseases. To achieve this, we will use all our expertise to realize your pharmaceutical solution. Why do we think we can support you? With over 100 and analytical development, as well as biopharmaceutical manufacturing, we deliver outstanding solutions that ensure maximum flexibility, uncompromising quality, and full regulatory compliance for our customer products. The advantage of this, to bring a pharmaceutical solution for life is our world-class end-to-end solution from process development through to clinical and commercial manufacturing enables seamless integration at every stage. We have the expertise to realize projects for cell culture and microbial-based proteins and enzymes, antibodies, virus-like particles, viral vectors, oncolytic viruses, vaccines, cell and immune therapeutics, and biosimilars. Who are we? Since October 2024, we have been 60% owned by the SK family, which ideally complements the existing Glocke Group family. As CEO, Dr. Sally Cho and Dr. Ulrich Wallay work as team to better meet customer needs. This resulted in multiple awards over the many years. We are, since the last 4 years, we have produced more than 4,500 GMP batches. And at our site, we have 12 US FDA inspections, and we are approved by EMA, FDA, and Visa. And since 1993, we have invested more than 600 million euros for innovative technology and production. Today, I would like to focus on the 5 highlights in the current analytical challenges for the release and characterization of viral vectors. But this is only a small excerpt, knowing that there are many other important aspects that we want to cover in upcoming webinars. First, I'd like to briefly discuss next-generation sequencing, which is increasingly being used to detect adventitious viruses in pharmaceutical products, especially in biologics. Following its inclusion in the ICH Q5 and FAMOI 2641 and increasing acceptance by the authorities, this analytical technology is increasingly being used. My colleague Stefan will address this topic in more detail afterwards. To illustrate the benefits of NGS technology, I would like to cite a few examples that have provided important insights into customer seed material. This enabled timely responses and prevented the wrong approach with repeated production. In general, a complete in-depth characterization in seed material is crucial to the success of failure of your product. This slide shows the quality issues we encountered during the incoming good inspection of seed material from our customer. We detect with our NGS platform incomplete transgene with deletions, insertions, point mutation, which may lead to impairment of the functionality. Also, that the transgene was inserted in a wrong direction and the flanking regions are not as expected. Furthermore, we detect a lack of genetic stability, which leads to losing the transgene or attenuation site. In the worst case, a full replicative virus will be generated. Also, impurities with parental wild-type vector is a topic. For example, for modified Vaccinia ankara virus, we find the wild-type vector. Adventitious virus stasis is very important and always in the focus. For cell-based product, Mycoplasma have also a high impact, and we have found Mycoplasma hyorinus in one product. Therefore, comprehensive testing, or as in the guidance of our industry expressed, extensively characterization of a starting material is one of the most important things for which NGS and current PCR methods are best suited. In contrast to Sanger sequencing applied in the past, NGS with millions of reads give a possibility to dive in deep in sequence analysis and to identify subpopulations. This guideline expresses very well what I described in the previous slide, but FAMOI 514, gene transfer medicinal product for human use, also places great emphasis on genetic and phenotypic characterization. Detailed information are also described in several other guidelines. In this chapter, 532 deals intensively with the characterization of vaccine seedlots. This requires, among other things, genetic characterization, including nucleotide sequence analysis, for which NGS technology is ideally suited. In addition to detecting attenuation, replication deficiency, stability of the insertion site, orientation and insertions and deletion should be tested. Genetic stability of a vaccine seed to a passage level comparable to a production batch should be demonstrated. Unfortunately, we occasionally shown that this genetic stability was not present. Regarding the investigation of genetic stability, our strategy consists in the viral vector over 5 passages analogous to the production process, whereby process-specific regularities must be assessed by a risk-based approach. Another example from a field of ATMPs is adventitious virus testing in production cells. A customer commissions us to test its master cell bank, which is used as feeder cells for culturing and conditioning NK cells for adventitious virus testing. The cell culture was carried out by adding tested human serum obtained from a blood bank. Using our fully validated NGS-based platform, we were able to detect sequences of 2 adventitious viruses. In the case of TORC viruses, a literature research and inquiries at the blood bank revealed that it was a known pathogenic virus with a prevalence close to 100% in the population. Therefore, the finding could be considered as non-critical. The second finding was significantly more critical, as it involved a human pathogenic retrovirus, XMLV. Using our follow-up strategy, we are able to demonstrate that it was a replicative virus using the PERT, Product-Enhanced Reverse Transcriptase assay, which detects reverse transcriptase activity. After initiation and out of specification, the cell bank was rejected as part of a further testing conducted there. Given the advantage of a known specific approach with the NGS platform, it is all the more important to have a follow-up strategy. This includes, for example, the development of a test method with stringent quality criteria, software for in-depth analysis, or the results and the predefined risk analysis. Furthermore, we have a high level of expertise with find customer or product-specific solution. Furthermore, we have supporting methods such as PERT, long-read sequencing by third-generation sequencing, and many different cell culture models conducting infection studies. Now, I'd like to move to a microbiological topic, namely endotoxin. The specification value is very low, especially for ATMPs and especially for ophthalmic application down to 0.1 endotoxin units per mil, which was a challenge and have to be measured using a standard curve with a limit of detection of 0.01 endotoxin units per mil. In general, endotoxin is still predominantly determined using the classic limulus amybicid lysate via the cascade shown here, with readout either by clotting turbidometrically or chromogenetically. The major disadvantage is that up to 20% of the animals died during the lysate extraction process, as shown on the left side on the left on this picture. As part of the initiative to avoid animal testing, we will switch to recombinant factor C, especially for new products in early phases. Unfortunately, the pharmacopoeia are not yet harmonized, and therefore, according to the USP, a comparative study is required. But when we see the seemingly happy animal in the image on the right, every effort should be made. Another aspect regarding endotoxin is the low endotoxin recovery value, which refer to the masking of endotoxin by components in the product. Specifically, chelators, surfactants, and/or proteins can cause the supramolecular structure, as shown in the image here, to change, preventing factor C from being activated. Authorities name mainly a demand from the FDA, therefore view the low endotoxin recovery effect as a potential risk, which may underestimate the endotoxin content in the product. Low endotoxin recovery studies are therefore required. The PDA technical report number 82 gives guidance for the low endotoxin recovery studies and is very helpful for this. The study design generally depends on the product and its production process. A design with 5 days at the maximum time point is shown in green on the right side of this picture. Now I would like to return to classic biological topic, namely titration to determine the content of replicative viruses and the comparison between the traditional method and that of flow cytometry. On the left side, you can see the classical approach, in which feeder cells are cultured and then infected with a viral vector in a dilution series. The readout consists of visual inspection of a cell monolayer for plaques, which only become visible after secondary infection when the plaque is large enough. The disadvantage of this approach is the subjective evaluation and the lack of data integrity, as the plates are discarded after evaluation and no digital data is generated. In contrast, flow cytometry-based titration is based on analysis at the single cell level during primary infection. Evaluation is performed objective by counting infected cells, and test duration ranges from 48 to 4 days. Digital data are available due to automated evaluation. A comparison table is shown on the right. You can see that the titration using a flow cytometer is significantly faster from in the mean 7 days to 2 days, and better when the accuracy is much more better and the CV much more lower. With a new generation of flow cytometer viruses down to a size of 80 nanometers can be directly characterized. This is ideal for high throughput analysis of process samples. Furthermore, a number of other analytical possibilities are opened up, such as determination of ratio between infectious and total particle concentration, determination of full empty ratio, determination of sample aggregation, very important aggregation, and expression analysis, because you can here multi-style method, implement a multi-style method. The topic of full empty increasingly becoming a focus, especially for AAV vectors, but also for analyzing defective and other viral particles. We use a combination of HPLC, SEC, and miles detector for this purpose. A key challenge in the production of AAV vectors is to maximize the number of full particles in the drug product. Other forms, such as empty particles, are considered product-related impurities that have a massive negative impact on efficacy and pose of safety concern. We decided to use HPLC-SEC in combination with the 18-angel miles detector to establish the method. Once the running conditions are optimized, we will use the UV 260-280 ratio as a GMP release assay. The other one is the other methods for detection of empty full was used for cryo-TM, analytical ultracentrifugation, the gold standard, and the ratio of genome titer and capsid titer. This comparison of various empty full methods show that SEC-miles and SEC-UV methods are very comparable to the gold standard of analytical centrifugation, ultracentrifugation. In contrast, the ratio between capsid titer and genome titer have pure accuracy, and with 0.9% of full particles in contrast to 65 and with a gold standard 71. This innovative approach is also for our viral vectors such as HSV possible. Rapid analysis is essential in cell and gene therapy, and speed is also a major issue for traditional viral vector and vaccines. Therefore, emphasis will now be placed on tests that have a long test duration, such as testing for mycoplasma and mycobacteria. Mycoplasma and mycobacteria are among the most important tests for viral vectors produced in host cells, alongside sterility and adventitious viruses. And nothing is more worse for a biologist or biotechnologist than contamination with mycoplasma, because the contamination progresses slowly and remains undetected for a long time. With the increase in clinical tuberculosis cases, the detection of mycobacteria is once again becoming more of a focus, especially with regard to contamination that can be traced back to an infected operator or gaps regarding environmental contamination. At IDT, we have developed our own dPCR for the detection of mycoplasma, fully considering the requirements of a detailed European Pharmaco Chapter 267, including extensive validation. The method also complies with the USP Chapter 63 and the other ones. This allows us to reduce the test duration from 28 days to 1 day and meets the requirement for sensitivity of less than 10 CFU. And the method also detects unculturable mycoplasma in contrast to the culture methods. With our proprietary dPCR-based mycobacteria detection method, we can detect a wide range of mycobacterial strains, including non-cultivable ones. The sensitivity is less than 50 CFU per mil, and the test time is reduced from 50-60 days to 4 days. A comprehensive or comparative study with the culture method has been successfully conducted, but product-specific validation is required. We recommended obtaining scientific advice from the authorities for the use of this PCR method. We learned about the benefits of NGS for foreign virus testing and other applications using examples, learned about the conversion of an endotoxin test to the recombinant factor C, and the low endotoxin recovery issue, delved into the possibility of flow cytometry for virus titration and characterization, gained an insight into the benefits of HPLC-seq analysis in combination with a Miles detector for determining empty-full AAV vectors, and examined the topic of fast-track analysis for mycoplasma and mycobacteria. IDT has innovative solutions for your challenges for these analytical topics, and for virtually the entire panel required for the release of viral vectors. And now I would like to hand over to my colleague Stefan Borutsky, who is a proven expert in NGS. As a bioinformatician, has also played a key role in the development of our pipeline. It's your turn. Thanks.
Thank you, Dietmar. Welcome, everybody. In the second part, we will focus on NGS and its use in the area of adventitious virus testing. I would like to focus on these 4 key points. I will first give you a quick introduction to the experience IDT has gained with the NGS technology and about the challenges we faced until the technology was ready to be used for adventitious virus testing. Next, we will briefly touch on NGS and adventitious virus testing in context of a changing regulatory environment. This includes acceptance of the method by the authorities. its addition to the guidelines as well as an overview of the current situation. Afterwards, we will come to the main topic of today's talk, IDT's virus testing workflow, outlining not only our wet lab process, but also all the additional work required to set up this system, such as qualification of sequencing devices, IT infrastructure, analysis pipeline, and up to the validation of the entire system. And last but not least, I will focus on that validation topic and different aspects that should be considered. IDT gained its first experience with an NGS technology in 2012 with Roche GS Junior system based on pyrosequencing. We have carried out initial investigations to confirm the identity of various materials like vaccine, virus seeds, or virus harvest production. The device was fully qualified and was ready to perform for release testing of commercial products. But due to the exit of the Roche sequencing system, IDTs had to change technology. A first MySeq from Illumina was purchased and qualified. The existing methods were transferred and adapted to the new system. New applications such as testing for genetic stability across different production stages or the investigation of specific single nucleotide polymorphism of attenuating vaccine viruses came into the focus of IDT's test panel. Testing in the lab and producing raw sequencing reads is one thing, but analyzing this with suitable methods another. IDT has set up its own IT NGS infrastructure to offer suitable and customer-specific solutions for analysis as well. In this environment, we design and validate our internal Adventitious virus testing pipeline called Labrador, according to the GAMP5 requirements. In 2021, our first fully validated virus detection method was ready for use under GMP conditions. With the use of the method come new requirements regarding the evaluation of the results. To confirm, further investigate real positive fits and possibilities of follow-up investigations, we tried to use long-read sequencing with the Oxford nanopore technology and transcriptome sequencing to identify potential viral mRNAs indicating viral replication. Due to the increasing requirements and recommendations in the guidelines, like a minimum read count per sample, we decided to implement the NextSeq 2000 to increase the sensitivity and the possibility to use different test options, like non-targeted sequencing with the maximum amount of reads per sample. Currently, we validate version 2 of our Adventitious virus test with different adjustments to improve the output. For example, the control system or the speed of the analysis pipeline. Let's take a closer look at some of the relevant guidelines, since these form the basis for all of our method development and adoptions. Two of the guidelines refer to the possibility of testing the absence of Adventitious virus using NGS. Since the end of 2023, the final version of the ICH Q5A in revision 2 was published. The new chapter 2641 in the European Pharmacopoeia describes the use of NGS for the detection of viruses. With regards to the new ICH, NGS is encouraged as a replacement for in vivo assays due to its breadth and sensitivity of virus detection and the limitations of the in vivo assays. Furthermore, this allows the introduction of NGS without a systematic head-to-head comparison. So, for the in vitro testing, NGS may be used to supplement or replace the assay. This addresses general limitations of the in vitro cell culture assay, like assay interference, toxicity, or the lack of effective neutralization of the vaccine virus. It is important to note, of course, that NGS comes with its own limitations. For example, it cannot differentiate between infectious particles and non-infectious viral sequences. Therefore, in case of positive results, these should foremost be investigated in an additional test system like a cell culture assay. In the European Pharmacopoeia, a new chapter has been added. The chapter 2641, High Throughput Sequencing for the Detection of Viral Extraneous Agents, describes the technology, method, and the possible workflow using NGS. But in the moment, it's only a draft. The new chapter outlines the different steps of the HTS workflow regarding the source of viral nucleic acid, possible sample material, pre-treatment, nucleic acid extraction, post-treatment, library preparation, and in basics, the bioinformatics analysis. In addition, Chapter 2641 also provides guidelines for NGS method validation, including recommendations for the selection of spiking material and the evaluation of the relevant characteristics for NGS. Now to today's main topic, IDT's Adventitious virus assay. This is a high-level overview about our method. I would like to split it in two parts. At first, the laboratory work until the sequencing procedure and saving raw data on an archive. At this point, the bioinformatics analysis starts. The reliable detection of adventitious virus requires a very good database, a well-structured analysis pipeline with multiple workflows to detect known and unknown viruses, and a very good understanding of how to evaluate sequencing data. A final report with the results is produced at the end of the analysis. Positive results represent DNA or RNA from viruses and must be further investigated to detect any potential infectious viruses. The information of the detected virus can help in the follow-up examination in terms of cultivation with the best host cell or to build up a specific PCR for further investigations. Now to the details. At first, our laboratory method. We can start with different materials like cellular or virus samples like virus harvest. We split them into a spiked and non-spiked sample. The virus spiked sample is used as a method suitability control and is an indicator to estimate the sensitivity with the specific sample metrics. We use a nuclease digestion pre-treatment step to reduce free cellular DNA to increase the ratio of virus DNA in comparison to DNA from the production cell. This step can be very different depending on the sample material. Additional options are different centrifugation steps to concentrate bioparticles or a chemical treatment to reduce background nucleic acids. After the extraction of the nucleic acids, the RNA and single-stranded DNA are transcribed into double-stranded cDNA. The library preparation is carried out with a standard kit of Illumina. At this point, it is possible to perform a targeted or untargeted virus testing. The use of hybridization probes leads to the enrichment of certain areas of the viruses, and so to a sensitive detection of a big range of known viruses. The non-targeted approach detects a broad spectrum of known and unknown viruses using a comprehensive database with viral sequencing diversity. We are using for the sequencing process Illumina's MySeq and NextSeq 2000 devices. In general, any sequencing technology can be used, but in the context of viral detection, generating more reads in a sequencing run may increase the probability of detecting the presence of extraneous viruses. Our bioinformatic pipeline includes initial processing of the raw data and different quality checks. The virus detection is performed by our own software pipeline called Labrador. At the end, our spike controls are evaluated and the final report generated. In case of positive results, a laboratory-based follow-up investigation is needed to assess whether the detected virus is associated to a real infectious virus. In context to the European Pharmacopoeia, EDT's approach includes the following points. As you can see, not all possibilities are covered, but it's not necessary. There's not only one way to detect extraneous viruses using NGS. There are certain aspects that need to be considered, like a good control or validation strategy, a bioinformatic pipeline that detects reliable virus sequences in a dataset with more than 1 billion raw reads. There's no matter which approach is chosen. For mapping or aligning reads, a reference sequence or reference database is needed. The FDA provides a reference virus database called RVDB for that purpose. This virus database forms the core of IDT's database. The RefSeq sequences of the RVDB, NCBI viral RefSeq, and the standard Kraken RefSeq database are clustered and generate more than 18,000 unique center for the taxonomic classification system. The diversity of the mass of different strains are added with the complete clustered RVDB. The final IDT database covered over 1 million virus strains. While the font of the picture is a little bit small, you can appreciate that both workflows illustrate the complexity of the analysis pipeline. The figure of the left shows the workflow of the whole analysis, starting with the storage of raw data in a long-term archive. Only quality-checked sequencing runs can be used in the Adventitious virus test data analysis. Labrador detects all viral sequences in the FASTQ dataset and provides a detailed report. This report is used by an expert to evaluate all positive hits and gives the final release with a summary report. The right side shows detailed workflow of Labrador. We use different databases like the IDT clustered RVDB, a protein-based database of the RVDB, and host cell genome databases to perform different classifications, mappings, or alignment steps. The yellow marked workflow depicts a quality trimming and host removal of reads that cannot be used for viral detection. More information about Labrador can be found in our paper. In addition, our IT infrastructure is based on 2 hardware servers for redundancy, and all virtual machines run with Linux operating system. The production system and the software pipelines are fully validated according to GAMP5 regulation for software. Regarding the ICH22 for assay validation, the Adventitious virus test method is considered as an impurity limit test. As such, specificity, the limit of detection, and in case of assay stability, the robustness are parameters to be assessed. In general, the specificity describes the extent to which other substances interfere with the determination of viruses. So it is the correctness of the identification. The spike viruses should be detected as expected, and non-spike samples should not show any virus detection. The detection limit is defined as a sensitivity and describes the lowest amount of viruses which can be detected. The robustness can be part of the validation or the development. That parameter investigates critical steps in the whole workflow, due to, for example, changing lots, material characteristics, and other. The spiking for the specificity and detection limit should be done with viruses representing the whole viral diversity in terms of structure, nucleic acid type, genome size, virus morphology, and chemical resistance. The following viruses are used at IDT as suitability controls and for the generic validation. All viruses are well characterized in terms of genome copy number, infectious titer, and the viral genome sequence. Since we use the control during the whole development and validation phase, we have good data on long-time stability of the viruses and have decided to continue using them and not to switch to the WHO panels as an internal test control. But for version 2 of IDT's Adventitious virus assay, which we are currently developed, we are planning to use a bigger WHO panel in addition to our 5 viruses to ensure comparability to other studies and companies. In terms of specificity, we checked the ability to detect our controls in different matrices and the correctness of the classification. In case of non-spiked samples, no virus sequences were detected. At first, in terms of NGS, the limit of detection is the analytical sensitivity of the method to detect low levels of virus presence in a matrix. Our samples were artificially spiked with our 5 control viruses to mimic different levels of potential contaminations. We spiked with 10 to the 3 and 10 to the 4 genome copies per mil sample material. In each of our sample material, it was possible to detect at least 1,000 genome copies per mil, but with a different amount of reads and therefore a different evaluation. Unfortunately, it is not possible to determine the detection level for all known detectable viruses. However, the LOD can be reported at the level all sampled viruses are detectable and indicate the worst case detection limit. IDT use a range of the detection limit for the specific matrix. In addition, depending on the number of detecting reads and the genome size, it is possible to calculate an estimated limit for other viruses with the same characteristics. These limits vary depending on the property of the samples which were tested. Samples with a high DNA background or a DNA vaccine candidate complicate the detection of other DNA viruses. Most of the sequence DNA is related to the host cells or the vaccine virus. For the robustness study, different materials were tested to show the reliability of the analysis with respect to variations in critical process parameters and sample characteristics. We tested raw materials like FBS, different cell banks, virus seeds, virus harvest material, and a virus mixture of the NIBSC from a study from 2015. Our results showed that the most critical parts are the sample extraction and the sample itself. Chemical components in the sample influence the extraction, and different amounts of background DNA reduce the amount of sequence reads referring to the contaminants. But all in all, it was possible to test all samples with a spike level of 10 to the 4 or better. However, since only a limited number of matrices can be tested, yes, there will always be new challenging samples. So our method is generic validated according to the use of specific sample materials. Equivalent matrices with no impact on the LOD can be directly tested or with a verification run. New matrices or matrices with an impact on the LOD need a new revalidation of the method. Nevertheless, our spike controls verify the LOD of each sequencing run and show the suitability of the method, the sensitivity, or problems during the laboratory work. To sum up my slides, the authorities open up step by step the acceptance of NGS for the virus detection and provide a good basis with the new guidelines. Using these guidelines as a starting point, IDT provides a fully validated NGS-based assay and supports end-to-end, if necessary, including in vitro follow-up investigation in case of positive findings. And last but not least, I hope I could convince you that NGS is very powerful technology for the broad and sensitive detection of known and unknown viruses. But the analysis of NGS raw data requires a high level of experience in bioinformatics to understand how the individual models work and to be able to be convinced in the final results. I would like to thank my colleagues working on NGS and the virus method. And thank you for attention. And now we're happy to answer your questions.
Thank you very much for this presentation. And just a reminder for the audience, to ask questions, you can send them via the questions widget. Just type them in this box and click submit. Please note that you can still send your questions after the webinar and they'll be answered at a later date. But it looks like we've got several questions already. Question number 1. What type of analytical tests are you offering in detail?
Yes, and a good question. In principle, we established a broad panel of analytical capabilities for release and for characterization of viral vectors and viral vaccines according to the guidance, beginning with molecular biological technologies from classical PCR in combination with agarose G electrophoresis, by qPCR with different qPCR systems, droplet digital PCR by using the for this we have 2 Bio-Rad systems, and also the new one, the digital PCR from QIAcuity from QIAGEN, and also as mentioned here in the lecture, technology for NGS, which is more and more common for characterization and for adventitious virus testing. And also for the classical biological and biochemical expertise, we have here implemented all kinds of titration. We have a lot of cell banks for titration, also for infectious studies, and we have a flow cytometer implemented also for innovative titration. We have the possibility for Western blot analysis and also for detection of replicative competent viruses by the combination of biological methods with PCR, with a readout of PCR. We have classical adventitious virus testing in vitro here, and also for this we have different cell lines. And for microbiology tests, we have a classical sterility, bioburden, endotoxin. We have we can identify the germs by MALDI-TOF system, and also we have high expertise for low endotoxin recovery values. And for the chemical physical testing, we have a broad panel of HPLC with a lot of all kind of detectors. We have different mass spectrometry systems for characterizing of viral proteins, also for capillary electrophoresis and spectrometry, different spectrometer. We have DLS and so on, and also we are more and more automate our systems. For example, for ELISA, we implement the gyro system for impurity testing, BSA, for example, bovine serum albumin, And so, yes, I think we have a broad panel, but special analytics such as electron microscopy or analytical ultracentrifugation, we have a partner for this, which done for us. But yeah, I think we are able not only to produce, but also to fully characterize the viral vectors.
Thank you so much. And question number 2. What are the main analytical challenges faced in viral vaccine testing?
Oh, this is a good, but also a difficult question. But because it's very dependent on the product. And as you know, viral vaccines, which are produced in cell substrate, in different cell substrate, with supplements such as BSA or HSA, human serum albumin, and also coli4 and so on. It's a very complex matrix, and therefore we have to adapt often the methods for the special matrix to prevent any inhibitory effects on the methods. And for example, often the vaccine virus doesn't allow in vitro tests for adventitious virus testing because it's very heavy or difficult to neutralize them. And therefore we use, and this is one point why we introduced the NGS technology. We use this technology as a replacement of a classical adventitious virus testing in vitro and in vivo, and also replace all the specific PCR systems or PCR methods with all applicable for which is all combined in the NGS technology. Yeah, and therefore I think this was one challenge, the missing neutralization. The other one is the complex matrix, which makes necessary to adapt the methods which are possible because a lot of methods are developed by us in-house. And for example, for host cell DNA, we have our own assay, and we have the possibility to adapt the system. And for AAV vector, as mentioned in my lecture, the difficulty is the empty full, and especially the partly full, or the vectors which contain also the host cell DNA. And therefore, we use for this also a combination of NGS, or in this case, the third generation sequencing, to sequence the complete encapsulated DNA, and can therefore characterize the particle from the drug product. And yeah, and several others, but this may be too long to explain.
Thank you so much. And question number 3. How do adventitious viruses impact the safety and efficacy of viral vaccines?
In the case of contamination of vaccines, it's with adventitious virus, in my opinion, unacceptable. And since removal is usually not possible, because you have a vaccine, a viral vector, and you have a contamination with the virus, it's in the most cases impossible to divide or to purify this from the vaccine product. And therefore, yeah, in all specification are mentioned, no extraneous agents are detected, and therefore it's not allowed. And yeah, and therefore we recommend strongly to characterize the raw material which are used in the production, as well as the seed material, and the seed material, the virus seed material, as well as the master cell bank, to avoid any contamination from these sources. And yeah, as mentioned for the APMP product in my presentation, when the raw material comes from human, in this case from a blood bank, when you have risk-based assessed, and you have to be in tight communication with the authorities, and if they accepted this or not. And for this, intensive risk analysis should be performed, and keep in mind if it exists any safety issues from the virus which is found. In the case of talk virus, that was because the high prevalence in the population, there was no safety concern for this. And in this case, and in combination with a discussion with the authorities, I think it's possible to accept this. But in the case of retroviruses, in the most guidance, it's very critical, and therefore it's completely not acceptable that retroviruses are inside of a product, and therefore, if retroviruses were found in the product, it should be discarded.
Thank you so much. And question number 4. Can you explain the role of next generation sequencing in detecting adventitious viruses?
Oh, yes, sure. The role has increased as the authorities adopt the guidelines and also include new chapter regarding the NGS technology. So NGS is more and more tested in different studies and offers a new opportunity to increase the safety. In my opinion, it is a technology that must be used. The breadth of detection simply offers the opportunity to characterize study materials like cell banks or virus seeds more in deep. So in terms of purity, it minimizes the risk of problems in later project phases.
Thank you so much. And question number 5. How can the sensitivity and specificity of adventitious virus tests be improved?
In terms of specificity, you need a very good database. Yeah? All virus strains in your database can be detected. So, to increase the sensitivity, there are different options. As a first step, you can try to work with a targeted approach and use hybridization probes. But this will shift the ratio of DNA contained in the sample towards virus contamination. But you have to keep in mind that you are also enriched the vaccine virus in your tube. Otherwise, you can produce more and more sequencing reads. So more reads simply means more information and deeper look in the sample. But you need to be able to handle this amount of data.
Thank you so much. And question number 6. How does the NGS-driven AVT assay compare in terms of sensitivity and specificity to traditional in vivo and in vitro methods?
So various spiking studies were carried out in the EAPS network. One statement is that the detection limit for the in vitro assays varies depending on the virus species. Comparing to NGS, which only detects the nucleic acid of the viruses, it has a slightly lower sensitivity, but provides very consistent results for the mass of different virus strains. This means that it is possible to detect every virus with similar LOD, so also unknown and viruses that can be cultivated like BPV3.
Thank you so much. As we're nearing the end of our scheduled time, we'll have to conclude our Q&A session. Please don't worry if your questions haven't been addressed yet, as the IDT Biologica team will follow up with you. So that brings us to the end of our Q&A session and webinar. I'd like to express my heartfelt thanks to Dietmar and Stefan for the outstanding presentation and for making this session possible. To all of our attendees, please keep an eye on your email for instructions on accessing the on-demand version of this webinar. You can also find it on our website at www.pmi-live.com. Once again, thank you for joining us today, and I wish you all a wonderful day ahead.
Thank you.